Hello, welcome friends. Myself, Professor Shantaya Gurmat, Department of Botany, MGVC Arts, Commerce and Science College, Muddebiyal, Karnataka State. So today's class we discuss about plant tissues, part 1. In this plant tissues, today we discuss about introduction and history of tissue culture. First we discuss about introduction. Plant tissue culture is a technique of growing plant cells, tissues, organs, seeds, or other plant parts in a sterile environment on a nutrient medium. Next, definition of tissue culture. Tissue culture is in vitro cultivation of plant cell or tissue under aseptic and controlled environment conditions in a liquid or an semi-solid, well-defined nutrient medium for the production of primary and secondary metabolites or to regenerate plant. Next point, in other words, it is an experimental technique through which a mass of cells called callus is produced from an ex-plant tissues. The callus produced through this process can be utilized directly to regenerate plantlets or to extract or manipulate some primary and secondary metabolites. Plant tissue culture refers to the cultivation of a plant cell which normally forms a multicellular tissue. When grown on agar medium, the tissue forms a callus or a mass of undifferentiated cells. The technique of in vitro cultivation of plant cells or organs is primarily devoted to solve two basic problems. Next one is to discuss history of tissue culture. The principles of tissue culture were evolved in 1838 to 1839 in cell theory advanced by Sleeden and Scaven. 1902, Haberland proposed a concept of in vitro culture. 1904, Hanning cultured embryos from several cruciferous species. 1922, Colte and Robbins successfully cultured root and stem tips respectively. 1926, Wendt discovered first plant growth hormone indole acetic acid. 1934, White introduced vitamin B as growth supplement in tissue culture media for tomato root tip. In 1939, Gouthard, White and Nobecoat established endless proliferation of callus. Next one is 1941, Orbeek was first added coconut milk for cell division in Datura. 1946, Ball raised whole plants of Lupus shoot tip culture. Next one is in 1954, Moore was first break callus tissue into single cell. Skoog and Miller gave concept of hormonal control like auxin and cytokinin of organ formation. In 1959, Rinnett and Stewart regenerated embryos from callus clumps and cell suspension of carrot. 1960, Cooking was first to isolate protoplast by enzymatic degeneration in cell wall. 1960, Bergman filtered cell suspension and isolated single cells by planting. 1962, Murshinge and Skoog developed MS medium with higher salt concentration. 1962, Kanta and Maheshwari developed test tube fertilization technique. Lastly, in 1966, Stewart demonstrated totipotency by regenerating carrot plants from a single cell of tomato. Okay, thank you friends. We meet in the next video. Thank you.